the second talk of this morning session. Can we have attention, please? Is Angela Sirigu, so please be seated and close the door in the back. Someone, you can start.
No,
okay, thank you. Angela Sirigu is the next speaker. Go ahead, Angela.
Thank you. Can you hear me? Yeah? Okay. Thank you for inviting me. And I was attempting to come here since 3 years, so finally I made. So thank you for the invitation. So today I'm going to talk about varieties of movement representation in the human brain. I work mainly with humans. I'm starting now some work with monkeys, but mostly what I'm going to show you is with humans, and mostly patients with brain lesions. And since, I would say, 12 years, I start doing some experiments in the surgery room. So that's what I'm going to talk about. So as you know, there are several brain regions that are involved in executing or representing movement. And the idea in our lab is what all of these areas are doing for movement representation and movement execution. What are these areas controlling? What kind of mechanisms are implemented in each of them? Of course, I just put some of them, the one that I'm going to talk about, but there are many others. So here we don't have a basal ganglia, for instance, and other brain regions. So the idea was, for instance, here you have the motor cortex. The motor cortex sends motor command. For many years, it has been said that this area contains a somatotopic representation of our body, and movement are represented in a discrete way. So this morning we hear a John that is not the case actually, and I'm going to argue the same thing. And then you have other brain regions, which are the parietal cortex. I studied for many years the parietal cortex patients with lesion in these areas. And I am always surprised the way in which these patients behave, because they can still execute the movement, but yet they don't know what they are doing, they don't know what are the intentions. So there are all of these sophisticated mechanisms that are implemented in this region. Then you have also the CIMeC. You will see that this is a very interesting area too. And we stimulate the area, and we got very strange results. And then you have the cerebellum that I'm going to talk first about. And the cerebellum, I have to say that I didn't have a special interest in the cerebellum. This is a very complicated area for us. When you look at fMRI studies, you have the cerebellum, whatever the subject is doing, you always have the cerebellum that is activated. So I think it's a problem for me. And so we went with very simple hypotheses with the cerebellum. So all of these studies that I'm going to present to you, they are done in the surgery room. That is, these are patients with brain tumor. And when the brain is on view, we perform stimulations, okay? And all of these studies, they are being performed in collaboration with Dr. Motolese, who is the head of the neurosurgery unit. And for many years, for 2 years, I went to the surgery room alone with my students, and then 2 years later, Michel Desmourget, who is here actually, joined my group, and I am very happy to let him go. So basically, what we are doing is very simple. We stimulated, and that's what Penfield did too. But in addition to what Penfield did, we recorded EMG signals, okay? Here you have an example of one patient. For instance, this patient has a tumor in the temporal cortex, and he's ready to go on surgery. So her head is placed in the stereotaxic device, and this patient performed the surgery awake, okay? So here, for instance, she's awake. So when the brain is on view, we perform the simulation, okay? So this is the simulation probe that we use, and usually the area that is stimulated is not the area where the tumor is located. Because the surgeon has to do a bone flap that is large, what we do is we stimulate the area close to the tumor area, but basically we assume that the area is intact. The area that we are stimulating is intact. So what we can do is that we stimulate at different intensities. For instance, it ranges from 1 milliamp to 8 milliamp. It depends on the region. For instance, M1, you need just 1 milliamp to trigger a response. The parietal cortex, you need to increase the stimulation. And then we record, we can do several things actually. We can stimulate it, put a grid in another area and look at the connectivity, for instance. And also, we can, so this is the time course of what we do. The patient is at rest. We perform the stimulation. Then, for instance, let's say a stimulation in M1. Then you have an evoked movement, and this is the motor evoked potential. Usually we put electrodes in a whole body to see whether we have response in the ipsilateral and in the contralateral side. Also, recently with Michel, what we did is we are searching, okay? Most of the studies, they are exploratory, okay? We are searching for sensory signals. We want to see whether, for instance, motor areas or areas that usually they never trigger movement receive sensory signals. So we stimulate at the periphery. We stimulate, for instance, the hand. We evoke a movement. And then we have a grid that you have here. And we record a signal, a somatosensory evoked potential. So let me tell you a little bit of what we did with the cortical stimulation in the cerebellum. So as I said, the cerebellum is an interesting area, but there are many hypotheses about this area. For instance, we know that after lesion, after degeneration of the cerebellum, we have what we call cerebellar ataxia. Patients, they have a problem in coordinating movement, reaching a target, and so on. Even focal lesion may give some kind of motor impairment. Yet, some people have said that perhaps the cerebellum is not involved in movement per se, but is more involved in, for instance, sensory discrimination. So we started this experiment asking ourselves, so if we stimulate, if we put an electrode in the cerebellum and we stimulate it, are we going to get movement? Can we trigger a movement? So this is a very simple response. And the other goal was, how is the body organized within the structure? We know that there is a somatotopic representation in M1, and we also know that there is a body representation in the cerebellum. And some fMRI studies have shown that this representation is not somatotopic. So we want to investigate this issue. So we had in this study 20 patients. We used increasing stimulation intensity. And we record, as I said, EMG signals. So here you have the kind of response that we have after electrical stimulation in the inferior cerebellar lobule or in the superior one. And you have here, for instance, this spot trigger thumb flexion. Okay, so they are very specific movements, selective movement. And here you have wrist flexion. And here you have the map that you obtain for the whole body. And for instance, we observed head and neck response here. And interesting, upper limb response, they are everywhere almost. They are in the superior and the inferior sector. And most of the lower limb response were found in the inferior cerebellar lobule. But the point that I would like to make today is that when you stimulate the cerebellum, we have, as I said, very selective muscle response. For instance, so these are all adult patients, okay? And in our sample, we had also very young kids from 2 years old. And we compare the kind of stimulation that we obtained in adults with the kind of stimulation that we obtained in kids. And the picture is completely different. This is a simulation in one adult patient, and you see that in an adult, the patient is 19 years old. The patient had a very selective response in the extensor. So let me show you this video. This is a video from one patient. So the patient is sitting because most of the surgery they are doing sitting. So look between the eyebrow. You see the movement, this very selective movement, okay? You have to be aware that if you want to move this muscle if you want to have a voluntary control on this muscle, you can't, okay? But we were able to evoke this movement with cerebellar stimulation. Now, if we look at what happened after cerebellar stimulation in kids, what we found is that contrary to what I show you in adults, we have very activation of different muscular groups. And most of the time, the activation, they are bilateral, not only unilateral. So we have a complete different picture. For instance, the same spot, okay, in the adults trigger a very selective muscle activity, but in the kids evoke a different kind of muscular activity. So why is this? The study is still going on, but we observed that the age at which there is a switch between several muscle activation and selective muscle activation is age of 10, okay? So we know that cerebellar output to the spinal cord or other stuff such as the basal ganglia becomes increasingly selective during development. And this leads to more selective body movements. And we know that also children, when you ask children to perform a movement, you can have movement of the limb that has been requested to move, but also movement on the other limb. That's what we call synchinesis. And we know that this clumsiness vanishes after 10 years of life. And on the other hand, we also know that in adults, lesion in the cerebellum lead to mirror movement. When you ask patients with this kind of lesion to move the right hand, they also move the left hand. They just perform like children. And they also show the synchinesis. And we know that the patients remain with this kind of synchinesis for a long time. So in our view, these findings show that this organization, okay, of selectivity of movement, organization of movement selection emerged probably during ontogenesis and is fully attained in adult. And probably the cerebellum has to do with the implementation of this kind of mechanism of selection. So one mechanism that we have hypothesized for the cerebellum is that the cerebellum is involved in movement gating. Now, if we look at what's the pattern of EMG activity that we get after stimulation of the cerebellum and after stimulation of M1 in the motor cortex, again, the picture is very different. They are stimulation for 2 adults, and you see here the selective thumb flexion movement that is evoked, and here you have upper limb, large upper limb movement that is evoked after stimulation in the motor cortex, which leads to the view that if the cerebellum is implementing selectivity for movement execution, the M1 is implementing movement synergies. Okay, we went to investigate this kind of movement synergies in M1, and we observed that, like Penfield has already described, in M1 there is a lot of somatotopy. And in a few years, several years ago, I don't know if you are familiar with the study by Graziano. Graziano published several results in monkeys where he investigated the effect of cortical stimulation in the precentral gyrus. He actually stimulated close in M1 and also in the Primordial Cortex. And the interesting find is that, contrary to the common view that M1 represents sample movement, Graziano triggered after stimulation very complex, not only very complex movement, but also postures. For instance, here you have the monkey has different the defense movement, reach to grasp movement, exploration of the peripersonal, the lower space, and so on. And then we have also another kind of synergies that is hand-to-mouth movement. This kind of movement, we were very interested in this kind of synergies because when you stimulate M1, well, first of all, it's very common in humans, but when you stimulate M1, you get a lot of movement with independent hand movement and independent mouth movement. So the goal in the second study that I'm going to show you is whether we could find spots in M1 that trigger both hand and the mouth movement. So in this second study that is already published actually, we had 26 patients and 7 subjects were also kids as usually. We have a lot of kids actually in our pool because there are a lot of tumor in the posterior fossa, and so I mean we can stimulate it because of the clinical outcome that cortical stimulation can have in these patients. So we had 7 subjects were younger than 10, including 4 that were younger than 4. So we stimulated 144 sites, which is huge, and 47 produced isolated motor response of the upper limb. That's what we expected, what Penfield has already described. For instance, here you have, so this is M1, and you have the layout of the different response that obtained in M1 for adults in blue and the children in red. And you will see that, so these are all the response, whole limb, arm, hand and the wrist. And this is the mouth response. And as you can see, all of these response, which are independent, I'm calling them independent hand response or upper limb response. All these response, they are in where we expect to see actually, what we call the hand knob, as classically described by Penfield and other people. And here we have the mouth response, where, and also again, is in the more ventral sector of M1. But we asked whether intermingled with this kind of response, we could also find some kind of hand-mouth synergies as described by Graziano. And actually, we did. Here we have a spot here in one patient. He's an 80-year-old patient, evoked movement, concurrent movement of the hand and the mouth. So the patient starts not only, we have only independent movement of the hand, but the hand moves in synchrony with the mouth. So you have the hand going toward the mouth, and the mouth starts to open. So this, what is interesting is that this response, they are intermingled. They are very close to the, so this is the layout of this kind of response in adults and children. And this response, they were located near to the one that, the spots that evoked independent hand or mouth response. We did another experiment where in the same patients, in the same session, we want to see whether this kind of hand movement response receives sensory signals from the periphery. And what we did is that we took all the spots that evoked hand movement alone, and we stimulated either the hand or the mouth. And the idea was to see whether the spot that is triggering hand movement receives signals from the mouth or from the hand. And, okay. And actually, this is the case. So we have here some hand motor and mouth sensory synergies. All the spots that evoke hand movement, when we stimulated the hand, we didn't record any sensory signals. However, when we stimulate the mouth, we evoked mouth sensory signals. In this case, we have a spot that responds to a hand movement here, and the same spot triggers a sensory signal when the mouth is stimulated. So we have here, contrary to the first kind of motor synergy, we have in the first one were hand-mouth motor synergy. Here we have hand sensory mouth synergies. And here we have the distribution of this response. And surprisingly, we were surprised because we were expecting to find this kind of response also in the distributed, at least within M1, in the hand area and in the mouth area, but the response were mostly located in the hand area. So to summarize this first set of results, we have independent motor mouth response, hand motor response, and we have also hand sensory mouth response and motor and mouth response. So within M1, we don't have only the representation of sample motor response for the hand and for the mouth, as it has classically been described, but we also have more complex representation involving different body parts like the hand or the mouth, and this representation can be bimodal, such as integrating not only motor signals but also sensory signals. Now we are in our ongoing studies, we are going to search for the other kind of posture that Graziano has described, but I think it's going to be a very long study. And we think that why M1 needs to have this kind of complex postures already represented. We think that these postures may subserve some specific needs from birth. For instance, the babies can bring the hand to the mouth even if the motor system is completely immature, and yet this movement can be done with very high precision. And one hypothesis is that there may be different sites in M1, one controlling some kind of motor prior, motor posture, and this kind of repertoire may be activated from birth, and other kind of sites that are controlling more fine skill movements. And it's interesting because this kind of dichotomy of movement representation mirror what Peter Strick has recently presented in a PNAS paper, where he injected the rabies virus in monkeys, was looking for M1 projection, and what Peter described is two kinds of spots in M1, one deep in the sulcus that he called new M1, and the new M1 has cells that make direct connection to motor neuron, and old M1 that is more in the surface, which corresponds probably to what we stimulated here. And in this old M1, the integration mechanisms, they are made at the spinal cord level. Okay. I think the question they are at the end, right? The question they are at the end. Okay. Now, I want to switch to another aspect of movement, which is the one that I studied actually for many years. And this, movement, so we trigger movement, sample movement. Sometimes in some areas, the patients execute a movement, they are not aware of this movement. And there you have other areas where the patient other area that never trigger movement, and yet the patient feels that has performed some movement. So we think that this area, this kind of sensation comes from activity in the parietal cortex. So I studied for many years the parietal cortex, and one of the hypotheses that was shared with other people is that this area, more specifically, area 39, the angular gyrus, is implementing some kind of sophisticated mechanisms such as sensory prediction about movement, forward modeling, and more recently we made the hypothesis that intention signals may be represented specifically in this area. I published two papers, one in 99 where I showed that when patients are performing movements, sometimes they are not aware that they are the agent of this movement. And more recently, in 2004, I did another study where I used Libet paradigm. I guess you are all familiar with Libet paradigm, which is, I have to admit, a very weird paradigm. And in this setup, we ask a subject to look at this clock and to perform different kind of judgment. The subject has to wait until the clock makes a first turn, and then he has to report where, what time was at the time of movement was performed, when he had the intention, or when he heard a sound. So this is the time course. So the clock appears, the first turn, and then there is a variable time where the subject is free to perform a movement whenever he wants, and he has to report these three different time of judgment. Now, we don't know exactly what is going on here, okay? So that's why I put dotted line, and presumably, there is some kind of decision that is made during this time. In this study, we tested different group of patients, control subject and two different group of patients, patients with cerebellar lesion, because they had movement, it was impaired in this patient, and the patients with parietal lesion. And here you have judgment, so here 0 is movement onset, and here you have the judgment for M, okay? I didn't put here judgment for S for the sounds, but it's like M judgment. Patients, they are like a control, very accurate. And here, patients like cerebellar patients and parietal patients, like parietal patient here, they are very accurate at the time of occurrence of movement. But when it comes to report when intention was made about this movement, cerebellar patients, like control, anticipated time, the onset of the W time wanting to move was very early, about 300 milliseconds before movement. But parietal patients reported time of intention very close to movement, as if they were waiting for feedback, sensory feedback to occur to make judgment about their own intention. And the interesting point about this study also was that not only there was this shift of intention in a patient toward the end of movement, but lesion, I had 6 patients in this study with parietal lesion, and all of them, they are very selective lesion, and the lesion, this is the overlap of the lesion were in the angular gyros. Okay? I had the left and the right patient, left and the right parietal patient, and in all of them, the spot that was lesion was the angular gyros. Now, so we made the hypothesis that perhaps in this region, they are representing some kind of intentional signal for movement. That's what we call a site of movement intention. Recently, there have been arguments that movement intention may be represented in the supplementary motor area. And you probably know the study by Itzhak Fried, who did intracranial recording in one single patient, and he recorded the urge to move, to perform a movement in the contralateral limb of the stimulated side. So in these two cases, you have a stimulation of the parietal cortex, where intention to move is impaired, and here you have a stimulation of the supplementary motor area, where patients report the urgent need to move. Now, so here, in a recent study, the group of Passingham, they performed an fMRI study where they used also Libet paradigm, and they reported that in accordance with what Fried was saying on the role of SMA in movement intention, they reported that when normal subject, they are performing intentional movement, they are made this kind of estimation about intention to move in Libet paradigm. What they found is that activity in SMA was increased compared to when the subject was judging onset of movement itself. So to disentangle between these two hypotheses, we performed a study in 2009 where we directly stimulated the parietal cortex of 7 patients that were under local anesthesia. And again, we record the EMG signals. And we asked the patients at the end of the stimulation what was experiencing. And here you have the kind of stimulation that, so this is the tumor. And here you have 2 spots that have been stimulated in this patient. And what we record is that after parietal cortex stimulation, mostly the stimulation, they were in area 39, because we were interested in this area, but we also recorded this kind of response at the border between area 39 and area 40. And what we found is that different spots in area 39 trigger the intention, the desire to move. So the patient report, I want to move my chest, I want to move my hand, I want to move my lower limb, and so on. So here we have this kind. One thing that is interesting is that when we increase the stimulation intensity in this parietal spot, the patients believe that the movement really occurred. It was not a desire anymore, but the patient had the illusion that he was performing the movement. So here we have an example of is a video. So in this is a patient that has been stimulated on the left parietal cortex, and he reported intention, desire to move the mouth, and after higher intensity, he reported that he had sensed something. He had the impression that he had talked.
Vous avez bougez pas.
J'ai envie de passer derrière dans la bouche.
De passer quoi? Bon.
De passer derrière dans la bouche.
Can you hear? We increase the stimulation.
2, 3, 4.
Vous avez bougé?
Oui, on va tout ça, la bouche.
Vous avez bougé la bouche?
Oui.
Micro?
So he had the impression to talk, and actually he asked me, what did I say? He didn't remember. Because we had the EMG recording, so the patient didn't move during the time of the stimulation. Then we had the occasion to test several patients with lesion with tumor near to the SMA, and here is a patient with stimulation. This is the spot that has been stimulated, is in the more dorsal sector of the SMA, but you will see that when you stimulate the parietal cortex, patient report desire, intention. It's the kind of movement that he believes to have executed as some kind of volitional aspect, and this is not the case after simulation of the SMA.
Tu as bougé, Mélanie?
Oui.
Ça t'a fait mal?
Non,
Ça t'a fait mal ou ça t'a fait peur?
ça m'a fait bizarre.
D'accord. Tu as...
So the patient in this case, even after the surgery, when when we asked her what was the kind of experience that she was feeling, is that the movement didn't belong to her. The movement was outside her body, even if her limb was moving. So we have here two kinds of experience. There is really different. The one reported by parietal patient that has really the character of an endogenous wanting to move, and here you have movements that the patient doesn't have the experience of having really sent a voluntary command. Okay, and by the way, we never trigger movement actually after parietal cortex stimulation. Contrary to SMA, with SMA at a lower intensity, you trigger an urge to move, but at a higher intensity, you trigger really movement. As in this case, movement is not perceived as belonging to herself. So I was very happy to see recently, so we argued that the angular gyrus in the inferior parietal cortex may be important in sending to the areas involved in executing movement, intention signals. And we were very happy in seeing that in the last issue of the Journal of Neuroscience, the group of Mark Hallett, Eric Besserman, and Liu He, they have just published this paper where they use a direct current stimulation, and they did a nodal stimulation in different areas, M1, parietal cortex, and the supplementary motor area. And they perform Libet task, now we are familiar with, and interesting, when, again, they also ask subjects, these are healthy volunteers, they ask healthy volunteers to perform 3 different types of judgment, S judgment, movement judgment, and intention judgment. And the stimulation didn't have any effect on this kind of judgment, but had a strong effect on when the subject had to report intention, time of intention, but not in all the brain region. For instance, this is the, oh, I'm sorry. So this is the sham stimulation. You see that after stimulation of the SMA, there is no difference between sham stimulation. And what is important here is that after stimulation, the parietal cortex and M1, time of intention was shifted even earlier than comparing to the sham condition and to the basal condition. So the conclusion is that it may be really mechanisms here, they are implemented in the angular gyrus related to intentional signals. And now we are continuing this kind of studies, and the idea is now is to map the parietal cortex and to search what kind of other response. For instance, are intentional signals in a different sector of the parietal cortex, inferior and superior. So far, we stimulated the superior parietal cortex and the inferior area 40, for instance, and mostly intentional signals are found here, at the border with area 40, but so far we didn't find any intentional signals in the superior, in area 7, for instance, but here we found, for instance, motor arrest. If we ask the patients to perform a movement, and while the subject is performing the movement and with stimulation occurs, the patients cannot perform movement anymore. So this is really a motor arrest spot. And then there is also another aspect that when we stimulate the periphery, the hand, for instance, or lower limb, we can get sensory signals in the parietal cortex. And these sensory signals, somatosensory signals, they are almost everywhere, I would say, but mostly they are concentrated here in the superior sector. Okay, so I think I'm going to conclude here. So I show you that there are different ways to represent movement in all of these different regions. Data from patients show that all of these regions, they are implementing different mechanisms. These different mechanisms may be different, that may be during ontogenesis, may be different, and One thing that I would like really to highlight is that the parietal cortex seems very important in one sophisticated aspect of movement, that is implementing intentional signal about movement and awareness of movement itself. Thank you for your attention.
Thank you for your talk. In Milan, we are actually doing similar studies mapping intraoperatively the motor system. So I was curious about your data, especially from the cerebellum. So the first part of your talk. I was not expecting to see any activation, direct activation of muscles from the cerebellar stimulation. And I was surprised also to see that from different spots in the cerebellum, you could reach the spinal motor neurons. Also, I was wondering which is the circuit that you're supposed to be underlying these responses, because different parts of the cerebellum are connected to different parts of the cortex, the cerebellum, the spinal cerebellum. So, in your opinion, which is the circuit that is underlying all the responses? Because I should expect differences in EMG activation when you are stimulating the cerebellum with respect when you are stimulating the spinocerebellum. And have you ever compared the activations of the EMG that you obtained stimulating the cerebellum to the one that you obtained stimulating M1, for example? And the intensity also, to me, when we are stimulating with bipolar probe 60 Hertz, it's very easy to reach, to get a response from M1, yes, but from non-primary motor areas, you need higher intensities. For example, from premotor cortex, you don't really reach any response unless you are doing a task. So I was wondering the intensities that you're using on the cerebellum. So this is my first question.
Yeah, can I answer? I mean, we use a different kind of intensity, okay? And of course, when we are simulate M1, as I said, with a very low intensity, you trigger right away the movement. This is not the case with cerebellum, okay? There has been also another result that we obtained recently, but perhaps Michel is here, he can talk also about. We did a connectivity study, and we simulated the cerebellum, and we got signal in M1. So probably there may be some, when we simulate the cerebellum, it's possible that there is a mediation of M1 also for this kind of movement that we trigger.
Which is the range of the intensity that you need in the cerebellum? Because M1 is 3 milliamp, 5 milliamp, depending on the cortical excitability during the surgery, if the patient is awake or is sleeping. But from the cerebellum, which is the intensity that you need to evoke a movement in terms of milliamps?
I think I have here, I don't know. Just 1 second.
Because high intensity could reach the pyramidal tract going down, for example.
Yeah, sure. I don't remember the intensity because Michel, he's actually doing the job in the surgery room. Do you remember the intensity? But be careful because patients, they are under the anesthesia, okay?
They are sleeping.
We cannot perform surgery in a cerebellar patient while awake, you know, because of the intubation problem that we have and so on. So
it's surprising, even more surprising.
Actually, even when we are stimulating the cortex, if the patients are under anesthesia, you need to increase stimulation intensity.
I know. I was expecting even higher intensities from the cerebellum, and I was just wondering if you could see any difference between the EMG profile when you are stimulating the cerebellum and getting a response from, for example, paravermis or from the lateral part of the cerebellar hemisphere with respect to what happens when you stimulate M1. Could be interesting to see if there is that connections or you postulate something different?
Well, I showed you before, I had the slide with stimulation of M1, EMG, but after stimulation of M1 and the cerebellum. And as far as we know, the stimulation of the cerebellum triggers always very selective muscle activity, very selective, comparing to M1. Of course, you don't have the same intensity, okay? Because as I said, if you stimulate at 8 milliamp, the M1, you trigger a seizure and it's not possible. So.
May I make a little comment regarding the representation of the hand and mouth on M1, so the second part of that talk. Just a comment. We are exploring that region and the premotor cortex anteriorly with high frequency when you can actually measure latencies, so you can be sure that you are stimulating M1 or the premotor cortex. And while with the low frequency you are evoking complex activity, so I would be careful in saying that is an overlapping representation, because with MEPs we are actually seeing that the overlapping representation of hand and mouth is more on the premotor side and not on, there can be a spread of activity or spread of a current. Just a comment.
Based on the subject MRI that we had, we were in M1. You know, we have a stereotaxic reconstruction of the sites that have been, of course it could be a spread, but when you go further toward the premotor cortex, and even locally, if it was an issue about the spread of activation, you could find the same response in the spot. But actually, this response, they were very selective to one single spot, okay? And as a matter of fact, I show you that they were intermingled between regions that evoked only end movement response. But I agree with you that there is the issue about the premotor cortex, and as a matter of fact, Graziano had a large region. He stimulated in M1, as far as he represented in the figure of his paper, and he went further to premotor cortex. And actually, most of the response that he reported for the different postures, they were, I think, in the premotor cortex.
This is a nice talk. I'm a little confused about your terminology for what you're calling the supplementary motor area. That wasn't, if you think about it, Woolsey defined secondary motor area in 1947 on the medial wall of the macaque. Later on, Penfield renamed the same general area, the supplementary motor area, exclusive to the medial wall of the hemisphere. That was followed later by cytoarchitectonic studies by Matelli and Lupino showing 6A alpha and beta were exclusive to the medial wall, followed by Zillis's work in the humans that it was exclusively in the medial wall, followed by imaging studies, starting with Roland with xenon that it's in the medial wall, followed by PET studies, our PET studies showing it's in the medial wall, followed by fMRI studies. So now you've got a lateral stimulation site that you're calling SMA. So I don't understand that.
Well, we can, unfortunately, we were not, of course, in the medial wall, okay, because we cannot stimulate it in the medial wall. Because when you stimulate the medial wall, well, we have a problem for the patient, as you know. So this is the more dorsal part, okay, of SMA. It's still SMA, the one that we stimulated, okay? But even if we look at the activation that were reported by Fried, for instance, who stimulated the medial wall, okay? He reported, the patient reported feelings of urge to move, which are the same that we observed after stimulation of this dorsal sector of the recall SMA, okay? So that's why perhaps I went too quickly to call this, to assimilate this with the SMA that is more in the medial wall, but the kind of response, they are not very different.
I'll ask a question, if I may. You find that there is this area where subjects have the intention to move. And if you increase the level of stimulation, they even have the illusion of moving, why don't they move? Why don't they move? If they have the intention to move, why don't they move? So what is the mechanism, or what role does this intention area play in the whole process of movement? I would have naively, not knowing much about this area, naively would have thought if I have an area that represents the intention to move and are stimulated, sometimes I should move. So why doesn't it move? And so what do you think is happening in this area, and what are the areas that interact with to permit movement?
So your argument is when I have the intention to move, I move because there is an intention, and logically movement should follow. Well, I mean, we stimulated, and then afterwards, okay, after the simulation is finished, we asked the patient what was the experience, what she felt, okay? I think that we explicitly instructed the patients actually to not move, okay? So I would be surprised, especially, for instance, one patient reported to have the desire to move the chest and to get up, okay? The head was in the stereotaxic system. I would be very surprised to see her moving and getting up, okay? But I think you are right. I mean, concerning, for instance, so in one case, the patient reported, I have the intention. Okay, of course, he didn't move. And then increased the stimulation intensity, and the patient had the illusion that he had to move. And actually, in this case, the patient didn't move, but had the impression that had to move. So in this case, the patient thinks that he had accomplished the movement, okay? So we think that what is going on in this area is some kind of sensory prediction about movement. So if we are activating intention signals, okay, these intention signals at higher intensity may be sent to motor areas, to the executive system, and transform them into real movement. So, but in this case, stimulation, what it does is that it creates an artificial activity, okay, within the brain. And in this case, the parietal activity, okay, was so high to provoke an illusion of movement. But actually, parietal cortex, in this case, was disconnected from what was going on in the premotor areas, for instance, okay? And this may have prevented the subject to really monitoring, okay, in time what was going on in the executive system. I don't know if I answered your question, but
I think I understand. But so could an alternative interpretation be that this area is not the intention area, but is the area that interprets actions? If it were the intention area, you might have thought that, well, then move. Alternatively, it could be that this area reads and interprets things as having been intentional. I mean,
there have been hypotheses also about this on parietal areas. Yes, of course. Yes, that's what we call sensory prediction.
I was wondering if you can comment, it's somewhat linked with Alfonso's question, about the interplay of somatosensory and proprioceptive signals in intentionality and ownership of movement.
Okay, I don't have any more of the slides. Can you have the slide?
Yes, just put it as a presentation.
No.
No?
But in any case, sensory signals, okay? We record the sensory signals in the parietal cortex, but they are not, most of the sensory signals, they are in area 7, actually, okay? We found also in area 39, but they are mostly in area 7. We think that, of course, there is an integration of these sensory signals. But the interesting point is that when we record a somesthetic signal in the parietal cortex, if the patient is awake, is not reporting any experience at the proprioceptive level, okay? So we recorded here in the parietal cortex sensory signal that not necessarily are, well, of which the patient is not necessarily aware of, okay? So we don't know yet, we don't have a clear hypothesis what is the relation between the intentional signals and the sensory signal, okay? But it seems to be, of course, it should be some integration somewhere, but so far, they seem to be in dependency. Laurie?
Thank you, Angela. I noticed in your data there's a hemispheric asymmetry in the parietal lobe, it looked like to me, in terms of the right hemisphere evoking a sensation or a feeling of desire to move the arm, and whereas in the left hemisphere you got these desire to move the mouth and the tongue. And of course that makes sense in terms of the known lateralization of language. But beyond that, I wanted you to speculate a little bit about how these intentions or desires to move might play into our spatial representations. Of course, we traditionally think of the parietal lobe as the place where you represent space, and there's a lot of data from lesions and et cetera that those lesions there cause spatial deficits. So could it be that the desire or intention to move the arm is more related to an extrinsic spatial representation, an extrapersonal spatial representation, or whereas these mouth and tongue movements are more closely related to intrinsic body-centered, configurational spatial representation. What do you think about this notion?
Personally, I think they are more linked to the body awareness, more than a spatial representation, okay? Yeah, I think that's more related to body.
So you think that this conjecture may be correct, that this hemispheric asymmetry in movement types is closely related to our spatial hemispheric asymmetries in spatial.
Absolutely. Yes, absolutely. Yes.
Thank you very much, Angela.
Thank you.
A reminder, please be sure to be back by 2 o'clock, because we have the presentations by the poster winners. And that, without taking away from the speakers, I think that's the best session. That's when our young people present their work. Please all show up at 2 o'clock.